Excellent. So next I'll be inviting Dr. Alba Grifoni, a research faculty / instructor at the La Jolla Institute for Immunology. Alba will be presenting using the IEDB in your research, Orthopox and SARS-CoV-2 examples. Thank you so much, Alba.
Lovely. Thank you so much, Nina. And as usual, very exciting to be part of the IDB workshop. And so far we have been hearing the beauty of the database and the different tools. And so what I would like to try to do during this presentation is to provide you an example of how you can use the IDB for your actual research. And so I'm going to provide two different examples, one on the Orthopox and one on SARS-CoV-2. And so the reason why we've been interested in the Orthopox is actually related to the Monkeypox outbreak. Many of you might be familiar with that. This is not a new virus, has been previously identified in the 80s, but this is the first time that we have a large outbreak outside non-endemic areas and has hidden over 75,000 different people in different countries. And so about the monkeypox, this belongs to the Orthopox virus families, and this is quite a large virus, a double-strand DNA, and comprise over 200 or more different orgs. And so part of the Orthopox virus families is also the very famous variola virus that was causing the smallpox infection. And as many of you remember, the smallpox have been eradicated through an active campaign of vaccination. And in particular, they were using the vaccinia virus strain for vaccination, which is actually quite similar also to the monkeypox virus. And so throughout the time, there have been multiple iterations of the vaccination, and partly that was due to the fact that the first generation vaccine, the Dryvax, that was discontinued in the United States in the 70s, has a very high efficacy against smallpox, but a very low safety profile in the sense that being this a live vaccine that keep replicating, it can be not safe for immunocompromised people that can have like very heavy reaction. And so because of that, there have been several iterations of this vaccine until the most recent one, which is the modifying vaccine Ankara strain, which is still a live vaccine, but is not replicating. And so while in terms of efficacy, we don't know what is the protection against smallpox in human population because that has been eradicated, animal models show very good efficacy so far. And now we have definitely improved the safety profile. And so the MVA and the ACAM 2000, I briefly mentioned that, but it's not that different from the Dryvax, have been currently licensed for monkeypox. So one of the biggest questions we wanted to understand is how our T-cell response are behaving in the context in general to vaccinia virus vaccination and monkeypox infection. And so in the past, the way to analyze T-cell responses was to perform in vitro infection with vaccinia virus and then measuring the T-cell responses. So we were wondering if you could essentially transition to this type of strategy that is time-consuming and also requires very high biosafety level to the generation of peptide pools that can actually speed up the T-cell response research in different labs worldwide. And so to do that, we have been designing peptide pools that were able to either detect vaccination or monkeypox infection. In terms of the first type of pool, we have been leveraging information already available in the IDB database for different viruses of the Orthopox virus family, assuming that considering the similarities between them, that could be a good starting point also potentially to detect the infection-specific T-cell responses. And so we did that. This is a tally of the query in IDB. As you can appreciate, the majority of epitopes, whether CD4 or CD8, and disregarding the host, comes from the vaccinia, the vaccine strains. And so we decided to design pools specifically for CD4 and CD8 T cells. For the CD4, we've been broader and we actually include multiple hosts with assumption that those can be also detected in humans. For the CD8 instead, we focus only on humans because we want to make sure about the HLA class 1 restriction, and that's what we were interested in. And so we also asked the question how much these epitopes that we look in the database was similar to the monkeypox. And we were happy to see that actually 96% of the CD4 and 80% of the CD8 were actually very conserved. And so now I'm gonna essentially show you the steps that we use in the IDB very quickly to actually extract this type of data. And so you already seen the IDB query, and so I'm not gonna go in explaining the different sections, but for instance, if we're interested in understanding what are the orthopox epitopes, particularly the CD4 epitopes, you could essentially click on the assay, make sure just to include the T-cell response and positive response because we want to look at epitopes only. You can select the organism, and for this specific query, we actually went broader. We wanted to check all the Orthopoxviruses. And then because we're interested in CD4 response, we select the class 2 MXC restriction. And in the context of host, as I was mentioning the data, we wanted to go broader, so we didn't select any specific host. So if you click on the query, the IDB is going to show you the different epitopes information in the different tabs. And so in the epitope tab, we see 345 epitopes. We have information about the epitope sequence, the different antigen pertaining to that, and the organism of origin, as well as how many references have been mentioned in these epitopes and how many assays have been actually independently show this as being an epitope. And indeed, if you look in the assay tabs, you can go much more into detail. And now you start having the epitope information linked to the specific reference, as well as the different information about the specific MHC restriction and the assay description of what has been run, as well as further details on the way the overall experiment has been carried out. If you instead are in general much more interested in what the reference are overall, and maybe you want to select specific reference for your interest, you can go to the reference tab, and that lists all the different references pertaining to your query, and you can extract the epitopes as you prefer. For the CD8, the picture doesn't really change. The difference in our query is, of course, because we're interested in CD8 responses, you select class 1 MXC restriction. And as I was mentioning before, in our query, we want to be selective in terms of the host, so we select human. And the results in terms of epitopes is essentially less than what I show you for a total response. The humans only have 249. And we extract this epitope information for CD4 and CD8. We perform a manual filtering just to remove epitopes potentially longer, and that could cause issues in the peptide synthesis. And then we design these pools. And so we were wondering, how does this pool are able to capture vaccine-specific response? And so to answer this question, we test this pool in a cohort of participants that receive the Dryvax vaccination. And so the cohort that I'm going to show you today are people that are healthcare workers, and they might have previously received the Dryvax vaccination either during childhood or for occupational reason. Maybe they received the vaccine prior to participating to this study. So in blue, I'm showing you the CD4 response in terms of activation on the left and in terms of production of cytokines, so functionality on the right side. And at the bottom graph, you do see CD8 response in red. The response is also shown in terms of magnitude and frequency of response that is represented by the different pie chart. So what is the picture that we observe? So first of all, I'm quite interested in the context of the CD4 response. We do see pre-existing immunity prior to vaccination that can come in either by childhood vaccination or older infection, therefore kind of suggesting that the CD4 response is indeed quite stable. Once you have the vaccination, you do observe a peak in the response of 2 weeks, and there you're able to capture essentially 100% or more, or roughly, of the response in terms of the T-cell response. And once you look at 6 months post vaccination, you do observe a significant decrease. However, still you have 60% of the response maintained, and in the context of the cytokine production, that response is even more stable and actually go to 94%. The picture that we observe in the context of the CD8 is quite different. We do not observe any pre-existing or very minor pre-existing response prior to vaccination. we still capture an increase in the response 2 weeks post-vaccination. The frequency of response is around 70%, which is what has been also showed in the context of other viral vaccination, for instance, in the context of SARS-CoV-2. But then once again, you do observe a steep decline at 6 months. And while that does not mirror the pre, and so we still have some CD8 T cell response maintained, the frequency of response goes as low as 20 to 30%. So all this data show us that essentially the pool that we design in the context of vaccination can be successfully used to actually track the T cell response induced after a vaccine. But in the context of the monkeypox, one of the questions we were wondering is, we know that these pools are similar to monkeypox, but it's also possible that then once you get the monkeypox infection, essentially your T cell repertoire broaden. And so to overcome that issue, we decide to use a different type of approach and design peptide pools that are specific to monkeypox. And so this approach uses, once again, IDB data, but really leverage much more into the analysis tools. And so that kind of give you a summary of everything that you can use while in a research project. And so we took, once again, the Orthopox epitopes, and then we essentially derive the sequence that are orthologs for the monkeypox. So that allowed us essentially to generate an inventory of orthologs peptides corresponding to the monkeypox. Once we had that, what we did was essentially to try to determine all the sequence what type of monkeypox protein they belong to. And that essentially allowed us to the following pie chart that I'm showing you right now. And that allows us also to have a range and a contribution of the different monkeypox protein to these epitopes. And so we decided to select protein that we consider to be immunodominant using the cutoff that each protein has to contain 5 or more of these epitopes. And that represents 67% of the CD4 response and 61% for the CD8, for a total of 19 and 40 different proteins. And then by doing that, we actually were able to apply T-cell prediction tools to essentially design a CD4 and a CD8 megapool that take into account the immunodominant protein for the monkeypox, but also factorize HLA. And so we make sure to use the reference set allele that were mentioned by Bjorn Peters before, so to make sure that these pools are done in a way that you take into account HLA that are frequent in multiple populations. And so ideally you can test the megapools in different geographical locations. So one of the questions we were wondering is, do we see in vaccinated individuals the capability to cross-recognize monkeypox? Because that bodes well on the fact that if you give a vaccine is going to help you out in actually minimizing the infection or even the disease severity caused by the monkeypox infection. And so that's exactly what we see in the context of both CD4 and CD8 T cell response. As you can see, the kinetics of response mirrors more or less what we already observe in the context of the vaccination, and both for the CD4 response and the CD8 response. Finally, and this is much more of a scientific point, we were interested in understanding the quality of the response, and we found that actually there was a multiple cytokine response, both when we look at vaccination and cross-recognition of the monkeypox pool, with an interesting prominence of granzyme CD4 T cell response, therefore suggesting maybe a contribution of CD4 CTL responses in the context of Orthopoxvirus T cell response. And so essentially, just to summarize this part, we have been using IDB in different ways. On one hand, to design pools that were able to detect vaccine-specific response, and we did that by leveraging previous information available for different orthopox virus. Then in parallel, we also designed megapools that were specific for monkeypox, and we did that by using orthologs of these IDB epitopes, and that allowed us essentially to select potentially proteins that are immunodominant for our T-cell prediction. Both pools, when tested in the context of vaccinated individuals, show a good T-cell response, particularly a stronger response and more stable response for the CD4, and an interesting broader pattern of reactivity with a large population of granzyme B positive CD4 T cells that is interesting to investigate. So this was just one example of how you can combine IDB and IDB tools for a research project. But now, I'm going to show you one point that we didn't necessarily address in that study, and I think is also important and one of the beauty of the IDB, which is the ImmunoBrowser feature that is embedded during your query. And so to do that, I'm going to just give you an example on SARS-CoV-2. So we have been using IDB and particularly information available in the IDB to try to understand what is the extent of the impact of the different SARS-CoV-2 variants of concern on previously identified epitopes. And so this is studies that have been done in the past 2 years, and essentially the results of these studies that the majority of the T-cell epitopes, no matter which variant you look at, are conserved in the majority of the case, and then there is a smaller fraction that is actually affected by the variant mutation. We then wonder whether these single point mutation were actually having an impact on the capability of the HLA molecules to present the epitope to the T-cell response. And so we did that by using T-cell prediction analysis. And what we found in the context of the CD8 is that 70% or more of the mutated epitopes are actually still able to be presented to the immune system, therefore suggesting that not only the conservation is pretty high, but actually even if you look at the mutation, it really depends where the mutation is hitting. And so essentially, when you get infection or vaccination, you have a portion of your spike protein or the entire virus that is presented with the HLA molecules to the T-cell response. The T-cell response react to it, and those are then therefore considered classical epitopes. When you have a mutation on a variant, in 30% of the cases, the mutation actually blocks the capability of that specific epitopes to be bound to the HLA. So no T-cell response, nothing you can do about it. But then a 70% of the response is actually still presented to the T cells. And so although the T cells, and particularly the TCR, might be very picky and unable to recognize the mutated epitopes, you can always have the possibility to generate a new T cell response with the mutated epitopes and kind of give you a balance on the reason why, in general, T cell responses are quite stable. There is another point in terms of the mutation, and is the fact that if you look at an entire protein, particularly the spike, the protein is not uniformly recognized at the T cell levels because that is dictated by the different HLA molecules, and those are one of the most variable alleles that you have available. And so essentially, if you combine the information on the T cell epitopes and you map them on the entire proteins, what you will see is the following picture. And so this is an example, and that has been done with the ImmunomeBrowser, and I'm going to dig a little bit more in how actually generate this type of figure. But essentially, if you want to look at the science, you're looking at the response frequency, so essentially how many times you have a positive response out of the amount of the time that has been tested, and accumulating all the different epitope information with the different restriction. And so the response is not uniform. You have some very high peaks that are considered to be immunodominant, followed by other regions that are actually poorly recognized. You can also appreciate that the pattern might not be exactly overlapping between the CD4 and CD8 T cell response, and that makes sense because, of course, the response is dictated by different class 1 or class 2 molecules. When you start looking at what is the impact of the variants, because one of the questions that can raise is, okay, the Omicron variant is starting to accumulating a lot of mutation. Is that starting to impact the T-cell response? So this is not just a matter of numbers, but it also depends where the mutation fall. And what you can appreciate is that actually the different mutation for the Omicron, and I'm showing you BA1, that was the original one that was used for this specific analysis, those do not necessarily coincide with immunodominant region. And that is actually providing also the reason why at the general population level, the T-cell response is still there to help you out, while you can still have in some specific case at the individual level some mutation on the Omicron that may impact some of the T-cell epitopes. And so how can you essentially recreate the same type of graph that I just showed you? And so I'm not going to spend much time on the CD4 query. You know the deal. Difference is that now you want to select SARS-CoV-2 as an organism. Once you go in your results tab, and particularly in the antigen tab, as Randy was saying, like ImmunomeBrowser really pop out at you because it's very useful and really allows you to combine the data and trying to understand at the antigen level what's going on. When you click into that on the ImmunomeBrowser, this is the type of output that you receive. So ImmunomeBrowser is going to show you this data in different ways. I'm going to go through them one by one. The first type of data that is going to show you is the response frequency. And so the IEDB calculate not only the response frequency, but also the confidence interval. So essentially, what is the range of your interval? And so this is what I show you here. You have a cursor, and that allows you to check per each amino acid position what is the interval, and your response frequency is essentially in between those two. You can also download the figure or also download information pertaining to the single amino acid position. Try to really have a feeling of what is the number of positive versus tested responses and what is the interval over there. So this is one way to look at the data. Another way to look at the data instead of in terms of response frequency is based on the assay. And so if you want to look, for instance, how many times a specific portion has been tested in how many different assays and how many times has been found positive and negative, this type of visualization provides you this information. Now, the two might be specular and quite similar, but they provide slightly different information. Once again, you can use your cursor to actually determine on each amino acid position what are the amount of negative and positive data, and you can download specifically both the images and also the information. Now, what if I am not really interested in looking just at the entire spike, but I just want to look at a specific region that is my favorite region? You can do that. So here the controls allowed you also to essentially fine-tuning your query on the residue position that you're interested in. And so IDB allows you to do that. Another point was, okay, so this is the frequency of the response, but how can I overlap this information with the variant of concern? And so this feature in the IDB allows you to select for the variant that you are the most interested in. If you do that, and I'm just going to show you the BA1 once again, what you can see is a very similar profile that I show you in my graph. This is updated with respect to what I show you because our current data. And what you can see is that there are different mutations in the Omicron, but the majority of those do not necessarily fall in the immunodominant region. But BA1 is kind of a story of the past. So is that changing on different variants? And so IDB allows you to do that because it has a large panel of different variants that you can visualize. And so, And so if we focus and we look at BA5 lineage that now is one of the most prevalent, what you can see is that the picture doesn't really change. You're still having a lot of mutation, particularly here, that is the RFVD region. And so that, of course, is correlated also to the fact that this allows the virus to enter the cells and the neutralizing antibody to block it. And so the virus tends to evolve much more in that region. But if you look at different portion of the protein, you do see that the mutations do not necessarily fall into immunodominant portion of the T-cell response. So with that one, I hope that I provide you a couple of examples of how you can actually use IDB for your research. The only thing I wanted to mention is the sciences team, and I show you a lot of data today, and those are really the results of a really fantastic teamwork. And I just want to make sure to thank the team at the LGI as well as our collaboration at the J. Craig Venter Institute. And with that, I also would like to thank my mentor, Alex Sette, that is going to take it away and dig more into the analysis tools. Thank you.
Thank you so much, Alba, for that wonderful presentation. I hope everyone enjoyed seeing how the IEDB can be put to use in a real life topical research setting as well. So thanks, Alba.